Doctor? Doctor?
Coming. Yes, please.
Doctor, help me.
What's wrong with you?
I'm having a problem understanding the meaning of my existence.
I am a specialized biologist and not a psychoanalyst. I'm afraid I can't help you.
And I am a specialized laboratory animal. I've given you my body, so now I'm also revealing to you my soul.
All right. I'll try and help you. Tell me, what's bothering you?
It all started when I began working in this laboratory. If I was a simple mouse, I could try to get by. And I could understand that the meaning of life is a piece of cheese. Now I'm a working mouse and I've got a full stomach. I don't have anything to do and you're continuously looking after me. But why did you choose me over all the other mammals? Why don't you experiment with elephants? I learn new words every day and I don't know what they mean. Science? Why science? Technology? Why technology? Biology? What's that supposed to mean? Biotechnology? Who knows what that tongue twister means? And if the meaning of life isn't a piece of cheese, maybe it is a word. One of those damned words.
Relax. In industrial societies, all workers face psychological problems. Your problem is relatively simple. You are suffering from intellectual shock. What you've learnt until now is not enough to face your new life. The remedy for your sickness is clear. You need more knowledge.
Will I get better, Doctor?
Yes, I think so. Just listen carefully.
See, if you try and think why a seed will always grow into a plant and a human oocyte into a human being, you will have to accept that both carry within them a specific plan or design for their development. In modern genetics, this design is recognized to be contained within a specific set of information-carrying elements called genes. The exciting discovery that revolutionized biology in recent decades is that this enormously complex genetic information that decides that a seed will grow into a plant and a human oocyte into a human being is encoded in a very simple molecule composed of only 4 different subunits. This is DNA.
In the early 1970s, DNA was the most difficult substance for the biologists to analyze. Today, DNA is the friendliest macromolecule to work with. It is now possible to excise specific regions of DNA, to obtain them in essentially unlimited quantities, and to determine the sequence of its nucleotides at a rate of several thousand nucleotides per day. Application of this technology enables the construction of novel modified or recombinant genes, which can in turn be cloned and introduced into suitable cells. Exogenous genes are often found to be transcribed and translated, altering the genetic character of the host in a designed way. Let us first consider some very important discoveries that led to the methodology of recombinant DNA. The existence of recombinant DNA methodology is rooted in the repertoire of enzymes that act on nucleic acids. Restriction enzymes are endonucleases that recognize specific base sequences in double helical DNA and cleave both strands of the duplex. Specific fragments of DNA formed by the action of restriction enzymes can be separated and displayed by gel electrophoresis. In many types of gels, the electrophoretic mobility of a DNA fragment is inversely proportional to its size, which is usually measured in base pairs. The mixture of DNA fragments, which is separated by electrophoresis, can be transferred to a nitrocellulose sheet. The positions of the DNA fragments in the gel are preserved on the nitrocellulose sheet, where they can be hybridized with a radioactively labeled single-stranded DNA probe. Autoradiography then reveals the position of the restriction fragment with a sequence complementary to that of the probe. A particular fragment in the midst of a million others can thus be readily identified. Fragments of DNA from any source can be amplified more than a millionfold by inserting them into a plasmid or a bacterial virus and then growing this recombinant DNA in bacterial cells. Bacteria are frequently employed as the host for maintenance and amplification of recombinant DNA molecules. This process is called DNA cloning and is aided by the combined use of restriction enzymes as molecular scissors and a different group of enzymes which join DNA fragments back into one polymer DNA molecule. The availability of many kinds of restriction enzymes and DNA ligases make it feasible to treat DNA sequences as modules and to move them at will from one DNA molecule to another. The ease of manipulations at the DNA level and the subsequent development of techniques for the introduction of natural or modified genes into somatic cells in culture provided the main methodological approach by which molecular biologists attempted to analyze gene structure and function. The so-called reverse genetic approach is generally applicable by creating specific mutations on genes outside the cell. A copy of an isolated cloned gene can be altered by recombinant DNA technology and then put back into a cell which now synthesizes an altered protein. Reverse genetic approaches have provided elegant solutions to central biological questions relating to gene structure, expression and function. Recent technological advances enabling the introduction of exogenous genetic information into the germline of whole animals, the so-called transgenic animals, provide the current state of the art in the analysis of biological systems.